Dear students, welcome to Indian Academy Degree College Autonomous. I am Professor Shubha from Department of Genetics. The topic for today's session is the cell cycle. The cell cycle is a sequence of events which occurs in the cell during which the cell grows and continues to divide. So therefore, the daughter cells are formed at the end of each round of cell division. The cell cycle consists of two major steps: the interphase, during which the cell prepares itself for the cell division, and the mitotic phase, during which the actual cell division takes place. The interphase itself consists of three stages: G1, S and G2 phase. During the G1 phase, the cell undergoes a lot of metabolic activities, which in turn helps the cells to increase its size, and therefore the cell growth takes place. And also the cell will prepare itself for cell division by duplication of cellular organelles and preparing different type of proteins which are required for the subsequent S phase. The S phase is the synthetic phase, during which the duplication of the genetic material takes place, so that at the end of the S phase there are two complete set of genetic material which are later distributed into the two daughter cells during the course of cell division. The last stage of interphase is the G2 phase, during which the cell continues to grow and further synthesize proteins which are required for the cell division. Coming to the second major step of the cell cycle is the mitotic phase after the G2 phase. So during the mitotic phase, there are two major processes which are taking place within the cell: karyokinesis, that is the division of the nucleus, and cytokinesis, that is the division of the cytoplasm. Mitosis consists of four stages: prophase, metaphase, anaphase and telophase. At the end of mitosis, there occurs production of two daughter cells from one parent cell in such a way that the genetic material which was duplicated during the S phase of interphase gets distributed equally in the two daughter cells at the time of cell division. In order to check that the entire cell division takes place accurately without any error, there are certain checkpoints in the cell cycle. There are three major checkpoints. The G1 checkpoint ensures that the cell is ready to enter from G1 to S phase. Once the cell proceeds from the G1 to S phase, it will commit itself to continue the cell cycle. The second checkpoint is the G2 checkpoint. The G2 checkpoint ensures that the cell has completed DNA replication or the duplication of genetic material without any error prior to cell division. And the third checkpoint takes place during the mitotic phase, that is between the metaphase and the anaphase, which is also called as the spindle checkpoint. This spindle checkpoint ensures that the spindle fibers get attached effectively to all the centromeres of the chromosomes, so that they are split equally and distributed during the karyokinesis. Therefore, at the end of mitotic division, we get two daughter cells from each of the parents. That's all for today's session. If you have any doubts, you may feel free to contact me. Thank you.